Okay, so first of all, of course, my sincere gratitude to the organizers for being here in person. It's a real pleasure to present our work to such a company of the outstanding specialists of the field. So today I would like to tell about some methodology which is not really conventional for this part of the investigations of toxinology, but maybe it will be interesting for you to see and maybe to use it further. So I'm representing here the area of my scientific interest, which is including the vipers of the old world. It's quite rare topic in these meetings. I don't know why. I think they are quite underestimated as a species, because even all European scientists now very much busy with the African toxins and South American and so on. But my interest is our local snake, which are including few species of the Macrovipera, Monzivipera, and Vipera families. And they are very interesting because quite endemic and rare. It's not about the Macrovipera libitina obtusa, which habitat is quite a big belt of the distribution from the Tunisia until the Kashmir. And it is including currently 5 subspecies, which is presented here, but I will talk mostly about the Macrovipera libitina obtusa. In taxonomy, I've mentioned by red color the species which are presented in our country. As you can see, the 3 of them are in the red book. They are quite rare snakes. And concerning the Vipera darevski and derivanensis, the venom of these snakes are very interesting also, but the low amount and the endangered species of the snake don't let us to work very much with this snake, with these venoms. So about the so-called West Asian blunt nose viper, Macrovipera libitina obtusa, its clinics is quite classical for the symptomatics of the viper envenomation. Usually the massive painful edema of the bitten limb extending to the trunk and other limb, blistering, myonecrosis, sometimes requiring, or quite often requiring the amputation of the bitten limb, thrombocytopenia, coagulopathy, hemorrhage, hypovolemic shock, acute kidney injury, and in severe cases, it could lead to the death. These pictures are not from our country because in our place, unfortunately, the investigation of cases are not presented very well. This is the pictures from Iran, Jordan, and some other countries where the snakes are presented. And unfortunately, sometimes they're still using the fasciotomy, which is not a good idea, I think, but still quite popular, at least in Iran, for example. So about the venomic profile, this is my, again, tribute to the Juan Galveto, with whom we've done this investigation many years ago. Further, we also repeated it with some Germany colleagues, and the most here are presented the proteomics, and most abundant and predominant toxin which are presented in these venoms are snake venom metalloproteases, PLA2, serine proteinases, and of course a big amount of different disintegrins. Later it was done the comparison between the venom proteomics of different Macrovipera libitina subspecies. And as you can see, the distribution of the toxins within the venom could be quite different from one subspecies to another. And sometimes it is only 4% of similarity, like between the obtusa and Transmediterranean subspecies. So in some cases, it could be totally different pictures of envenomation. Now, what we are fascinated most in these venoms, that separately, these toxins are very weakly toxic, and the fractionation usually are not giving us too much information about the impact of different components in some processes which are we usually see during the envenomation. So we've already many years working with other experimental scheme, which is designed like trying to understand the impact of different toxins within the venom, inhibiting them in the whole composition of the venom. So for this investigation, we've tried 3 different types of inhibitors, which are able to block the activities of PLA2, snake venom metalloproteases, and snake venom serine proteinases. Quite conventional inhibitors, very well known with their activities, and we are trying to see how the assay of the neutralization of hemorrhagic activity, which is usually used when we are trying to testing different types of antivenoms to see this efficacy. So inhibitor was incubated with equal volume of the venom solution and keep them for 30 minutes at the temperature of the organism, and then aliquots of 100 microliter were ID administered to group of rats. And as a negative control, of course, we used saline instead of inhibitor was used, but we also have had the positive control concerning the same without the venom. I mean, we try also the hemorrhagic activity of BPB, EDTA, PMSF themselves to be sure that they are not adding something in the hemorrhagic activity of this composition of venom with inhibited toxins. And the hemorrhagic lesion on the inner surface of the removed skin were observed 24 hours later. And then it's a usual measuring of the activities, which now we are doing not with the ruler, but of course with the computational imaging analysis. And the picture was quite interesting and even a little bit surprising. During the BPB and DDTA, when we are suppressing the activity of PLA2 and metalloproteinases, we have some slight inhibition of the hemorrhagic activity, about from 26 to 33%. But when we try to use these inhibitors in combined together, we have the bigger hemorrhage area than usually. So this is really interesting. We see that the area with hemorrhage increasing from 17 to 31% even. So we thought that it should be the third agent, which are mostly responsible for the hemorrhage. And of course, logically, obviously, we think about the serine proteinases. And when we try to suppress the serine proteinases within the whole venom composition, at this case, we see the quite obvious, again, the decreasing of the area of hemorrhage. For example, as a comparative reason, we can see also the neutralization of hemorrhagic activity by antivenom, which we obtained in our place. It's the ovine-derived IgGs. In this case, of course, it's 100%, but the suppressing of the hemorrhage by inhibiting serine proteinases is about more than 50%, which is also not that bad. But of course, in this case, we have a number of things which are not really satisfying us with the picture, because we see the problems with this area of hemorrhage, and we've decided to try some new methodologies which could assess these differences better. For that reason, we let's see how the hyperspectral imaging analysis could be useful for this problem. I don't know whether you're familiar with this technique, but just a couple of words to explain what is this. Hyperspectral imaging is a technique that analyzes a wide spectrum of light instead of just assigning primary colors to each pixel. The light striking each pixel is broken down into many different spectral bands in order to provide more information on what is imaged. So in this technique, it's combined the optical spectroscopy with optical imaging. When from every pixel, we have not only one color spectra, but the whole spectra which are possible to make like cube, they say like hyperspectral cube. Our technique is nuance, which is very good camera. It is quite mobile. You can take it to the field with you if it is necessary, or in some clinics. And it also could be combined with microscope in case if you would like to see the more deeper analysis of the picture, or even during the surgery, because now it's became more and more popular also in the surgery. Primarily, the technique was developed for the astrophysical, agricultural, some other problems, but now it's became more and more popular for life sciences also. So this is the working interface of this camera. You usually do able not see only the very surface, but a little bit deeper inside the skin. And also it is possible to compare, to put the threshold of the spectral line which is necessary for you, but you also can do some phone, which could be the skin color of the same rat or the skin color average from the different rats. And even more, you can also assess the hemorrhage from the surface under the hairs without removing hair. But again, we go to more conventional scheme. We are removing skin and then trying to see what information can give us the hyperspectral imaging. I should say that hematoma itself is not a very homogeneous thing. And in case of the interest of the spectral point of view, it has a lot of interesting, colorful information, because when the chromatophores of the different type of, let's say, conditions of blood are quite different. The chromophores of oxyhemoglobin is red, deoxyhemoglobin blue, methemoglobin is brown, carboxyhemoglobin bright red, melanin brown, biliverdin green, and bilirubin yellow is also presented in the simple bruises or hematomas, and of course in the hemorrhage also. Except of that, we also should say that the layered nature of the skin, they affect colors as a result of scattering properties that affect shorter wavelengths more than longer wavelengths, and can exaggerate to attenuate hematoma color perception depending on the depth of injury. So it is not only about the blood conditions, but also different helpers and different other secondary products which could be collected in the place of the hemorrhage. Well, after all this explanation, we've done the same scheme of analysis of the hemorrhage of the venom inhibited by BPB, DTA, and also PSMF, and analyzed all this ton of imagings of hyperspectral analysis. We've seen that in this case, of course, the increasing of the hematoma is not so significant. So it when we see by the image that area of hemorrhage is bigger in case of the inhibiting of metalloproteinases and PLA2. In this case, it's not significant. So maybe it's just not a big differences, or it's not a deeper injury than in case of just BPB or DTA. But the results with inhibiting of serine proteinases is even more visible in this case. So we can totally conclude that at least for this venom of Macrovipera lipizina obtusa, we would say that serine proteinases should be the most responsible for the hemorrhagic local tissue damage than all other predominant toxins, which are presented mostly in this venom. But again, I would like to say that in case of the increasing of hemorrhage during the inhibiting of PLA2 and metalloproteinases, we will continue this investigation, because we already have a number of other literature data that in some cases the significant, synergistical character of the work of the toxins not only able to enhance the work of each other, but also suppress. And in some cases, the PLA2 could suppress the inflammation for some reasons, maybe for spreading effect or some others. So we will continue to investigate this also. And I would say that the fourth component, which is also quite well presented in this venom, is C-type lectin-like proteins, which we will try to investigate further also in combined with all of this. And maybe it will be some new interesting results also. And I would like to say that hyperspectral analysis is a quite new potent instrument, but we will try to use not only in such kind of model experiments, but maybe also during the clinical analysis, if it will be possible for us in our place. Thank you for your attention. This is our team, and I would like to say thank you also to our colleagues Naira Ayvazian and Sergey Fedotov for the consultancy of the hyperspectral imaging, and all this was done also by the support of the Chan Zuckerberg Initiative, which is mainly devoted to the biomedical imaging. Thank you very much.